All right. So this is the continuation of the previous lectures. This is the second part. In the first part, we already learned about the hormones and how they've been synthesized, what target cells, what are the nomenclatures, okay? How do we name it based on the structure? How is the classification? How do we categorize the class 1 and class 2 hormones? As for today, we want to go a bit more deeper and understand what is the method of transportation. So you know that the hormones is constituted of two different types, okay? One is hydrophilic, means they can be soluble inside the water. In other words, they can travel at their own free way, in their own freedom, okay, without any issues or problems. The second one is the lipophilics. Lipophilics means they love to attach to the lipid molecules. It means that they are not water soluble. So they hate water. Lipophilics means hydrophobics, okay, because they have these two ends, okay, the hydrophilics one and also the hydrophobic one. So the lipophilic one, they love the lipid condition and they cannot travel inside the bloodstream readily, okay? They must ask from help from other people, okay, from other carriers or other helper, or what do we call other transporter or other transport protein. They need help for them to travel into our body before it can give effect. So today we want to go and understand what is happening, what is the way or mode of transportation that is available for these hormones. The first way of transportation is via plasma transport proteins. What is plasma? Plasma is the main constituents of your blood, okay, of your whole blood. Okay, we have around 55% of plasma in our bloodstream. Okay, another 45% more or less you can say it's constituted of your blood cells. Okay, it can be erythrocytes, okay, which is the red blood cells. It can be leukocytes, your white blood cells. It can be some other immune cells, okay. It can be other supporting cells too, right? But in this context, the hormones can travel in the plasma. Why? Because around 90% of the constituent of the plasma are water. Okay, so you can see that most of the hormones are water loving. They love to travel with the help of the water or inside the watery condition. Okay, so this is the basic what we call introduction to the different classes of the hormones. So the way that they can be transported here now, they are using the plasma transport protein. What is plasma transport protein? Plasma transport protein are proteins, okay, that are available inside the plasma in the blood vessel, yes, and then they can help to transport the hormones, okay, from the endocrine organs or endocrine cells. When the hormone is being secreted, they can get into the blood vessel easily. Okay, so they can travel easily inside the blood vessel. All right, so the in this slide, okay, I put it in this slide. The class 1 hormones are hydrophobic in chemical nature. So this is the hydrophobic one, okay? Not very soluble in plasma. Okay, they don't like this one. This class 1, they don't like the water that much. Okay, they love the lipid condition. Okay, so they are not very soluble in plasma. This is true for the steroid and also thyroid hormones. Okay, you know, previously last week we have gone through what are the different types of thyroid hormones. It can be thyroxine, it can be the 3-iodothyronine, Okay, it can be thyroid hormones, okay, and some other hormones. These are the lipid-based hormones. They have a very specialized plasma transport protein, right? Unlike the hormones that love the water, these class 1 hormone, they don't like the water, and then they need to use the help from the transport protein for them to get transported through to the target cells. Okay. All right, so they have a specialized plasma transport protein. This protein circumvent the solubility problem. Okay, it means that with the help of the transport protein, the hormone can be transported easily. Okay, regardless of the structure. For example, on the right hand side, you can see the lipid soluble hormone. Okay, the image on top, on the right hand side, you can see the lipid soluble hormones. They want to travel into the stream too, just like the water soluble one. However, here the transport protein comes into action. Okay, so the lipid soluble still can be transported into the bloodstream. Okay, so these proteins circumvent the solubility problem. Okay, in other words, they already solved the problem with hard to dissolve. After that, deliver the hormone to the target cells. Okay, there are various ways that these lipid soluble hormones can act on the transport target cells. Okay, hormone when bound to the transport proteins, they cannot be metabolized. They can only be transferred or transported, and this way can help them to stay at a very active state. Okay, they are not easily metabolized. In other words, they are not easily degraded. Okay, for the water soluble hormone, yes, they are easily degraded, but for the lipid soluble hormones, it's quite difficult to dissolve them or to degrade them, and then it contributes to the very long lifetime, okay, inside the blood vessel. They can stay there for a few more days, like maybe 1 week. But for the water soluble hormones, they can stay around 1 day, okay, maybe less than 1 day, maybe hours, right? So this is very important for you to know the differences between class 1 hormones, which are hydrophobic, and also class 2 hormones, which are hydrophilics. Okay, so the hydrophilic hormones, which is the class 2, they are made of the peptide structure. Peptides means the single units of the basic unit of the proteins. Okay, you can have one peptide, we call it one peptide chain, we call it monopeptide. We can have like dipeptide, we can have like tripeptide, we can have very polypeptide structure. Okay, when the amino acids start to become increasing in number and they attach together. So the hydrophilic hormones generally class 2. Okay, some book they put the hydrophilic class 1. Okay, the lipid soluble hormones class 1. Sorry, class 2. So it can be either. Okay, but there's only 2 different class. Okay, it should be fine. Whether, because if you see in this picture on the right hand side here, you can see the class 1A, the first one is the water soluble hormone, which is the peptide, and the right one is the lipid hormone. Okay, don't get confused. It's okay whether it's class 1 or class 2, right? But you need to know that there's only 2 different types of hormones. Okay, generally you can classify them based on the components. What are they made up of? Okay, the soluble one made up of peptide, the lipid one, okay, made up of the lipid structure. For example, okay, for the peptide one, you have the insulin growth hormones, adrenocorticotropic hormones, okay, TSH, thyroid stimulating hormones, they circulate in the free and active form and very short plasma half-life. Okay, why they have very short? Because they don't have any attachment to it. They are in a free form and they are simpler to be degraded. So that's why the insulin can be present in the body for quite limited time. All right. So when you consume something, okay, during the lunch, for example, the pancreas, okay, the beta cells of the pancreas start to secrete this insulin. And this insulin will enter the blood vessel and then take up all the sugar molecule, okay, the glucose molecules, and then they store this glucose molecule inside the target cells. Okay, this is very, very quick. Down here in the box on the table here, I've shown you the features, okay, the differences between receptors and transport proteins, okay, not the different type of hormones, right? So again, let us recapitulate. In this image, you have the peptide hormone. If you can imagine, this is an insulin, okay, secreted by beta cell of the pancreas. You can see this they excrete the hormones to the outer environment, and then this insulin will enter easily the blood vessel. After that, they will bind to the target cells. On the target cell, they must have the insulin receptors, okay, the receptor protein to detect the conformation that they have from the insulin. And after that, they can decide whether they want to go into the nucleus of the target cells, or they just want to give effect or some protein synthesis inside the cytoplasm, okay? So that's a very simple or straightforward way for the water-soluble hormones. On the other way, okay, lipid-soluble hormones, the receptors in nucleus or cytoplasm. You can see the receptor can be present in the nucleus, okay, not in the cytoplasm. And some other occasion, they can also be present on the cytoplasm. But the good thing here is that the lipid soluble cannot enter the blood vessel without the help of transport protein. And this transport protein will help the lipid soluble hormone to go to the target cells and bind to the cell surface of the target cells. And after that, this hormone will be inserted into the target cells, and after that they will recognize a specific receptor protein inside the nucleus. And after that, if the hormone structure match the receptor protein inside the nucleus, they can regulate the gene transcription. All right, whether you want to increase expression of certain genes, or you want to down regulate the action of the certain genes. Okay, well, you can choose whether you can be the activity of the target cells can be inhibited, or the activity can be stimulated or activated. So this is one of the way. A bit more complicated, means they have add up more cascade or more step inside the lipid soluble hormones, right? However, on the table down here, you can see the concentration for the receptors. Okay, when I mention the receptors, this is the differences between the left and also the right one. Okay, receptors in the water soluble are very low in terms of the concentration, but in the transport protein, the concentration is very high. Okay, inside the lipid soluble condition or way, the transport protein concentration is very high. You need more transport protein because lipids is not readily dissolved inside the bloodstream. You need a very high concentration to make sure that your body can compromise or can handle the increasing amount of the lipid hormones. How about the bindings affinity? In the receptors, okay, the first one, even though the concentration of receptors is very low, but the binding affinity is very high. What is affinity? Affinity means how well they can bind together. If you have like enzyme and substrate, you have learned this in matriculation center, or you have like a lock and key conformation. They are very specific, like this. My knuckles, okay, in contact with my palm, like very specific, means that very high affinity. They can bind immediately. But in the terms of the transport protein, there might be some misconfiguration. It can be like the hormones cannot fit into the receptor. Maybe the receptor is very big, so it cannot bind to it readily or specifically. So what happens is we call it as the low binding affinity. They can bind, but then after some time they can lose the hormones. Okay, they are not fixed like the receptors in the water soluble hormones. This will contribute to the binding specificity. As I told you earlier, receptors, if they bind to the hormones very tightly, so they are very specific and they can last very long. No, not long, but because they can be what we call, they can elicit the effect on the target cell very quickly. Okay, so like the lipid base, they need to take some time to, you know, make sure that the conformation is fixed. All right, and this will take some time. That's why the lipid soluble hormones are struggling. Okay, they will take some time inside our bloodstream to stay. Okay, before the target cell can give effect. Okay, so this is just an example. Hormone transportation. Okay, again, the second way is by transporting the thyroid hormones together with the thyroid binding globulin. So globulin is a global proteins as well. These are the helper protein. Okay, remember thyroid hormones are the steroid hormones. They need the helper, they need the carrier, they need the transporter to help them to be transported. So you can see they can 1/2 to 2/3 of the T4 or thyroxine. Okay, this T3, 3-iodothyronine, that has been secreted by the thyroid hormones in the body, okay, is in an extrathyroidal reservoir. What is extrathyroidal reservoir? It means that these hormones, T4 and T3, are most of them, okay, 80% of them, more than 75% of them are available outside the thyroid organs, okay? So they are available outside the thyroid because they have been secreted from the thyroid, most of them, okay? Once they have been secreted by the thyroid, they quickly go to the bloodstream and quickly go to the target cells. And to have this one, they need the thyroxine binding globulin, a special protein to carry the T4 and T3 into the bloodstream before they reach the target cells or the nucleus of the target cells. TBG is a glycoprotein. What is glycoprotein? Glyco refers to the carbohydrates or the glycan. It means that this is the combination of the glycan, all right, coming out, what we call synthesized from the or metabolized from the carbohydrate source and also the protein structure. Okay, why do you need the glycan or the carbohydrate together with the protein? Okay, there are various there are multitude of purposes, okay, the function of the glycoprotein, because if you have only protein without the glycoprotein, you lose the lipid side of it. Okay, you don't have any other activity or you cannot support the transportation of the protein, because the protein on its own are very fragile inside our body. So with the help of glycan, when they bind to the protein, forming the glycoprotein, it can help them to be transported further. Okay, since this is the lipid-based hormones. Under normal condition, the TBG the thyroxine binding globulin non-covalently bind, okay, nearly all the T4 and T3 in the plasma. Okay, now this glycoprotein wants to bind the T4 and T3 once they're been released by the thyroid hormone, by the thyroid organs, okay, by the thyroid, and then be circulating, and they will circulate inside the plasma. And T4 bind with greater affinity than T3. It means the specificity between T4 and TBG are more stronger, okay? They are more fixed together as compared to the T3. T3 is a bit simple structure and they might lose the conformation, okay? So they have lesser affinity with the TBG. All right, so down here, this is, you can read the plasma half-life of T4 is 4 to 5 times that of the T3. Okay, the plasma half-life T4 long lasting, okay, because they stay more, okay, fixed together, since this is a lipid hormone, so they can be circulated or can be transported further away, like from the thyroid goes down to your limbs, for example. Okay, however, maybe for T3, maybe they can just bind to the liver only. Okay, very short time, okay, very short distance only. Free T4 and T3, the bottom, okay, the image or the diagram in the bottom, I put it on the left hand side in yellow. You have the thyroxine binding globulin, the TBG, 99.9% binds to the T4. Okay, the thyroxine binding albumin, okay, albumin also transport protein. Okay, so you can find it in the white egg. Okay, this is important for the cell-cell, okay, communication if you want to transport the thyroxine and 3-iodothyronine. Okay, however, the TBA, okay, albumin, okay, binds to T3 only at 99.7%. Okay, but the thyroxine binding pre-albumin, the very small amount remain unbound. Okay, this is the very free, very they are not attached to any TBG. Okay, they are free, we call it free, they are not attached to any glycoproteins, so they can give effect immediately, because this free very low in terms of amount, and some point they are not in a active they are not readily what we call helping in transportation. All right, they can circulate inside in the body, but they can be degraded quite quickly because they don't have the glycoprotein or any other transport protein attached to them. Okay, so on the right hand side in the table, I summarize the T4 and T3 regarding their the concentration. Like T4 inside the plasma is very high, 8 microgram per deciliter. However, the percentage of total inside the plasma is very low, 0.03. Okay, what is this? This is the free hormones, because they are very high concentration attached to the glycoprotein, but the remaining are very low. I mean, this is the free T4. Okay, here they put 0.04 and 0.4. This is quite okay similar. Okay, 0.04. Okay, some books they put 0.03. So don't get confused on that. The half-life in blood in terms of day, T4 can last like maybe 1 week, 6.5 days. T3 can go for up to 2 days max. Okay. Hormone transportation. Okay, again, this is the third way how the hormone can be transported. This is glucocorticoid. They've been transported by corticosteroid binding globulin, hydrocortisone, or other name we call it cortisol or stress hormones, can also circulate in the plasma in a protein bound and free form. They can bind to the transport protein and they can also travel in the free forms. Okay, this is true in the case of the glucocorticoids. The main plasma binding protein is the alpha globulin. We call it as transcortin or CBG, corticosteroid binding globulin. They are being synthesized in the liver, and the estrogen can stimulate or increase the production of this CBG. All right, CBG functions to bind to the glucocorticoids. All right, after stimulation by the estrogens for giving the effect to the target cells. Again, this is the lipid-based hormones. CBG binds most of the hormone when plasma cortisol are within the normal range. Only, so there's an exception here, right? So the CBG can only bind to the cortisol, can only bind to the glucocorticoid when the cortisol level in the plasma are normal. Okay, under stress condition, if you feel stressed, then the CBG cannot bind to the glucocorticoids and you cannot get the effect after that. However, binding to the CBG is not restricted to glucocorticoids. There are some other corticoids can bind to the CBG. For example, here, deoxycorticosterone and progesterone can bind to the CBG with sufficient affinity to compete for cortisol binding. Okay, this is another example. CBG is not exclusive for glucocorticoids. They can also bind to the deoxycorticosterone or progesterone. Second one, CBG can also bind to the aldosterone. This is the most readily potent natural mineralocorticoids, does not have a specific plasma transport protein. So CBG also help to transport the aldosterone inside our body. Okay. The third one is gonadosteroid bind very weakly to the CBG. This is the steroid hormones, okay, for the sex organs. Okay, the CBG can also bind, but very weakly. On the right hand side, at the bottom, you can see in the table SHBG and also CBG. SHBG is the sexual hormone binding globulin. CBG is the corticosteroid binding globulin. You can see the concentration. This is the affinity as expressed in nanomole per liter. The hydrotestosterone, you can see in the SHBG, another globulin for sexual hormone. Okay, binding globulin, they can bind to the hydrotestosterone. Only 1 nanomolar per liter. Okay, the unit is down here. But the CBG, corticosteroid binding globulin, more than 100 nanomolar per liter can bind to the hydrotestosterone. Why? There can be many reasons. It can be due to the specificity, as I said, the binding affinity. If they match together perfectly, then more of the CBG can help to transport the testosterone hormone, can transport the dihydrotestosterone hormone, the estradiol or the estron. All right. So this is true for the CBG. However, in the case of the the case of progesterone, cortisol, and corticosteroid, the main transport protein are the SHBG and not the CBG. Okay. So this is a very specific sexual hormone binding globulin that have a perfect match with the progesterone, but not the CBG. Okay, so when I ask inside the MCQ or the multiple choice question in your final exam, what would be the transport protein for progesterone? Okay, what is the most available transport protein for progesterone? So the answer is SHBG. If I ask you what is the most readily available transport protein for testosterone, your answer will be CBG. Okay, here. The fourth one, This is the gonadosteroid, SHBG, as I mentioned previously. The same thing, okay? Most mammals have a plasma beta, not alpha, okay? Previously, we have the alpha globulin, but in the gonadosteroid hormones, most mammals have the beta version, okay? This is the subunit of the globulin. Globulin is not only one structure, but sometimes they have a subcompartments or subcomponents. They have an alpha, they have beta, delta, mu, whatsoever. Okay, so they have many configuration and many subunits. So in most mammal, we have most of them are beta globulin that bind testosterone with high specificity, high affinity, but limited capacity. All right. So if they match together perfectly, very specific, so they are very limited. Okay, because they come and bind, come and bind, come and bind. There are no loose structure or components that cannot bind. Okay, so this related with one that we have discussed, the SHBG, or in some other book they put it testosterone estrogen binding globulin. They are being produced in the liver. Production also increased by this hormone, sexual hormones, the estrogen, and also the hyperthyroidism. Okay, in the case of hyperthyroidism, this is also a mechanism for the transportation of gonadosteroids to give an action. Since HBG and albumin bind more, around 99% of the testosterone in our blood, only small fraction of hormones is available in a unattached form. Okay, so because they are very specific, then you can find only very few structure. Okay, only very few of the estrogen or gonadal steroid hormones that are available. Most of them, 99% of them, bound to the SHBG because they are lipid hormone. They cannot be transported. They cannot be active without attachment to this transport protein. Okay, so the figure below showing you the how to illustrate this in a diagram form. You can see from the hand side bioavailable testosterone. Okay, you can see low affinity binding, corticosteroid binding, globulin, CBG bound to the testosterone. Okay, you have the SHA, albumin bound testosterone, and then orosomucoid bound testosterone, lots of transport protein, but they are very low affinity binding. They are not specific. However, when these are being converted into the free testosterone, okay, unbound testosterone, testosterone that are not bound to any albumin or corticosteroid or orosomucoid, okay, the free testosterone can bind very highly specificity or high affinity to the SHBG. Okay, so this is the transportation. What happened if you have this free testosterone bound to any of these four transport protein? Okay, so this is the explanation, more explanation, what I have mentioned earlier. So regarding the SHBG, what are the causes of increasing SHBC, the increasing transport protein in our body? Okay, this is due to the increase in estrogen. You know, some people have diseases, or maybe they are not having any disease, but at certain period of time, their hormones are misregulated. They are not regulated properly, and then leads to the increase or decrease certain hormones. In this case, estrogen, what we call is not regulated properly, and increasing estrogen can cause increasing the SHBG concentration in our blood. And there are some other disorders that can also lead to the elevated SHBG. Some diseases can also lead to the low level of the transport protein, SHBG, inside our blood. Now we move on the action of the hormones. Last week we have learned about the hormone introduction. What are they? What are different types? Now we want to focus on the action. How we can trigger the action. What are the mechanisms that they can be transported into our cells? How quickly they are? How can they transfer the signal? Are the similar signal they receive to being passed on to the target cells, or the signal has been modified and then being reduced so that the action can be inhibited? So there are various ways. We call it the propagation of the signals. In other words, we call it the signal transduction. How the signal can be passed on via a different cascade of information before the action can take place. The hormone receptor interaction can cause the intracellular signal that regulate the activity of a selected set of genes. These signal transduction can affect the DNA. They can produce certain signals, and this signal is important for your genes to overexpress or to underexpress, or we call it increase in terms of the gene expression or downregulation of the gene expression. So these are all being innervated or being stimulated or being triggered by these hormones. So this complex, the hormones, when they bind to the receptor, they can create a set of signal, and this signal will tell the cell what to do. And these can change the production or can modify the production of the protein. It can be producing more protein or very less protein for the use of the cell, or maybe they can delay a certain activity or can make it quicker. These include the enzymes or the transporter or channel protein, right? So these are the key players. What are the components that involve in this activity? The signal can influence the location of protein in the cell and can affect the general process, such as the protein synthesis, the made-up of a new protein, okay? Cell growth, for the development of the cells. From one cell, you want to grow, split into 2 cells exponentially, then become 4 cells, and then become 16 cells, and then 32 cells, and then 64 cells exponentially, okay? So this is based on the hormones action, okay? Hormones signal. And also for the replication. Replication involve the DNA synthase, or what we call the mitosis, for example. These are also having the information or signal from the hormones that telling them, okay, when they bound to the receptor, they will tell the cells to replicate, okay, or increase or decreasing the certain expression of the gene. Of course, the hormone is not only the players. You need a help from other supporting players, such as the signaling molecules. This might include the cytokines. We have thousands of different cytokines, interleukins, IL, okay, in short, glucose growth factors, GF, and also some of these metabolites. They have different functions at certain time point, okay? So you need to know that these are the other supporting molecules to help the hormones in entering the cells and in packaging the information so that the target cells can give the effect. Excessive or deficient or inappropriate production and release of hormones and the regulatory molecules are the major causes of disease. Okay, if you have overstimulation of certain hormones, for example, the thyroid is producing more T4 and T3, then you might have the hyperthyroidism. Okay, if you're lacking the hormone inside your blood, you're having hypothyroidism. If you have more, if you have less insulin, for example, so it can lead to the hypoglycemic, or the insulin is not functioning well, you can have the hyperglycemic condition. So inability for the body to regulate a good amount or normal amount of the hormones can lead to these disorders, diseases. Many things can happen if you have this problem with the hormones production. This is the summary that I want to show you, between the different type of group of hormones. Okay, it's important for you to know the hormones can transport the signal, can transduce the signal, can give the signal to maintain our homeostasis. The stimulus can be challenge or threat to the organism, to an organ, or to the integrity of single cell within that organism. Okay, for example, the body is being confused. You are producing insulin, and at the same time, you're producing very high glycogen at the same time. Okay, so how do you want to handle the glucose level in your blood? Or you having T3, very high T3 and very high T4, and after that you are having, you know, very low T4 and then very high T3. So how the body regulate this? So this is important for you to know, that the step-by-step process should be passed, okay, should be perfect before they can carry out with the next step. All right, so this is just the example to illustrate this. You start with the signal and recognition of the signal. You can classify the hormones into two, then they can be released. We're either making a hormone plus the receptor complex, or generating a different types of signal. And then this signal is important at the gene level inside the nucleus of the target cells, or they can act on the plasma membrane of the target cells. In some other occasion, it can goes up to the translation. All right, you know, the DNA transcribed into the mRNA, mRNA translated into the protein, and then the protein will be translocated into some other cells. And also might be involving some protein modification. Maybe you want to add more protein or amino acid to the peptide chain. All right, so this will also happen in the group 2 hormones. All right, so the effect is a coordinated response. You have to pass certain criteria before you can pass on the information. If your body are attacking your own hormones, then you will have something like the cells are killing your own body cells, and this is very fatal. Okay, so for example, your cell are producing a protein that can kill the parent cell. I give you example here for the plasma, what we call it, a plasma based hormones. This is the hormones transduction that can affect the homeostatic or mechanism. This is involved, okay, the signal transduction, the way that the signal being coordinated and processed through this signaling pathway. Okay, there are thousands of signaling pathway that we need to study, and one of the most famous one is the insulin signaling pathway. This is an example of the condition, like inside the diabetic condition of the patient. You see, the insulin hormones released by the pancreas bind to the insulin receptor on the plasma membrane of the target cells. The target cells can be what? Liver, can be adipose tissue, can be the muscle cell, where they will help. Okay, the insulin will take up the sugar, okay, with the help of glucose transporter, and then keep it into the muscle or in the liver or in the adipose tissue for further use later on. If you deprive of energy and you are not having your lunch, you feel hungry, or during fasting, you cannot eat, you cannot have food, right? So that is where this glucose molecule will be converted into the ketone bodies for energy production in a form of ATP. ATP is like a fuel for your body cells to functions when you are deprived of energy. So in this example, insulin will bind to the insulin receptor, and after that, they will generate the signal. This is quite complicated for you at your step, but just to illustrate what are the things. There are lots of proteins, there are lots of factors, there are lots of supporting molecules that can help the transduction of the signal before they can give effect to the cell. So you see, in between, inside the signal generation, there are different ways and very busy and complicated way how they can regulate or activate a certain activity. For example, in the protein translocation, you can see where that is where the glucose transporter comes into action, insulin receptor, IGF-2 receptor. It can be translocated into some other location if they follow this pathway. But how about this pathway? Okay, from IRS1 to 4, okay, you have this PY, okay, and then after that go to this phosphate amino acid kinase, and then it can goes to increase certain enzymatic activity, and this is involving the insulin receptor, phosphatases enzyme, and so on. Some point they can also activate the gene transcription, and this is true for these molecules. All right, this component, the molecule GRB2 MSOS, I don't know what are they, okay, but I'm sure this is the signal transduction molecule that can be packaged, okay, that has the functions to conduct the transport the information to the target cells. All right, you don't need to know what are these molecules. It's not important at your stage, but it's just good to know that there are something happening in between before the effect comes into action. All right. Same goes here. This is the example. The lipophilic group 1 hormone diffused through the plasma membrane of all cells, but only encountered they are very specific affinity intracellular receptors. Okay, non-specific intracellular receptors and cannot bind these hormones, right? And this can cause the activation of certain activity. Did I miss someone? If someone is leaving the group, maybe you have class after this, yes or no? What time you're not free? Is it okay for us to continue? Yes, everyone.
Yes.
Okay, can we continue? Yeah. Okay, maybe sometimes people have the internet connection issues, but do not leave the class because I have the recorded version. You have to let me know if you have any issues, if you cannot attend the class, for my session. All right. I am your coordinator for biochemistry subject. For any biochemistry lecture, you have to inform me if you have any issues. Okay, either with Dr. Samsul, Dr. Nurhazira, Dr. Azmin. Okay, you have to let me know if you have any issues in attending the class. Okay, it's important for you to have a full attendance throughout the semester. Get back on what we are discussing now. The hormone receptor complex can be activated for making a certain action. All right, so this is the example by two different class hormones, the thyroid hormones and also glucocorticoid steroid hormones readily gain access to the cytoplasm of the cells. The glucocorticoid hormones, the solid triangle. All right, so the solid triangle is the glucocorticoid hormone encounter their cognate receptor inside the cytoplasm. Okay, the description is down here. This is only illustration just to show you again this involving a signal generation. You don't need to know what is TRE. You can learn. I have provided the description if you want, but just for you to know that this is not an easy process. This must go through a certain level of signal cascade. Okay, just a summary. If the information or the signals goes into the nucleus and start to give information, or certain transcription factor can bind to the gene, or certain promoter will be produced to bind to this energy, this information from the gene can be transcribed into the primary transcript. After degradation and modification, it can cause the leads to the production in mRNA. So this occurring in the nucleus. In other words, this is the transcription process. Outside the nucleus, when the mRNA is ready, okay, from the DNA to chain, this one you have only the ribonucleic acid chain. Okay, you have the only single chain, not double chain. Okay, from DNA, which is inside the gene, and then you have the RNA. This mRNA will be a messenger RNA. Okay, mRNA will be transported. So the name is messenger. It means that you need to package the information. You need to collect the information. You need to send the message, because this is not the final stage, right? Once this has been transcribed into a smaller information, this mRNA need to send the message that they receive from the gene, okay, from the mRNA into the cytoplasm. And this is where the final stage comes into action. We call it the translation of the mRNA into the protein. And then when they have the information in terms of what protein to be produced or synthesized, then you can start use the protein to give certain effect. For example, if you have a hormones that giving a signal that we are depleting amount of insulin, so you need to produce another insulin, new insulin inside the beta cell of the pancreas. So what the cell do? They need to go to the DNA, tell that we need more insulin, and then they will modify it into mRNA structure inside the nucleus. Okay, this is where the information being transcribed. Okay, and after that, this information will be messages. Okay, will be carried out or be transported outside the nucleus to make the protein. Okay, remember that the protein is being produced or synthesized inside the rough endoplasmic reticulum. You have the ribosomes. Inside the ribosome, where the amino acids be synthesized, whether you can produce a primary structure of protein, or you have a secondary structure, you have the quaternary structure, whatever structure you want. Okay, it's telling that, okay, this is the information that we have. Okay, we need to make a new insulin component. Okay, we need to make a new insulin hormones. All right, so this is the final stage, or the protein as a effect of this signal transduction. Not only for the new synthesis of new protein, but it can also the signal transduction can also meant for producing protein for activating a certain action or inhibiting certain action. Okay, not only for producing a new hormones. There are various ways, all right, various effects of the signal transduction. Now we move on to the carbohydrate metabolism. There are three types of hormones: epinephrine, glucagon, and insulin. So this is important in the case of carbohydrate metabolism. Maybe today you will have a class with Dr. Samsul, I presume, but this is just an introduction, okay, regarding the carbohydrate metabolism. The epinephrine, also called the adrenaline. They act on the muscle tissue to raise level of glucose on demand. You know, in the PBL, you have learned about the fight and flight. Okay, Gopal tried to run away from the stray dog and the muscles start to constrict. Okay, when the muscles start to function, you need to supply them with a good amount of energy. All right, so the energy come from the glucose that you are taking. Okay, we have mentioned just now the insulin that take up all the glucose and store it in the muscle tissue. This is what happening. Okay, this is what's happening. And based on the structure, this the adrenaline, the epinephrine, the structure is quite similar to the tyrosine. All right, so this is another amino acid. They are being released from the adrenal gland. Okay, where is the adrenal gland? You know the renal. Renal is referring to the kidney. Adrenal glands means they are the endocrine glands that are available on top of the both kidneys, both of your kidneys. What's the function? To produce or secrete the epinephrine. Okay, it's important for the fight or flight response. For your PBL, in the anatomy PBL 1, okay, maybe this week you have another trigger 2 to discuss fight or flight response. How the body handle the increasing demand of the ATP or the energy source, and how the signal has been conducted or regulated in preparing yourself for this response. All right, so we have reached this far. Hormones control carbohydrate metabolism. You know that this is similar that we have discussed previously. You have here the receptor that bound to the epinephrine, okay, in red, and it activates the G protein. Remember, we have lots of subunits of the G protein. We have the G alpha, G beta, and G gamma. And in turn, this G protein, when they are being activated, they can stimulate or activate the enzyme adenylate cyclase. What is this AC enzyme doing? Okay, adenylate cyclase enzyme functions to convert the ATP into the 2-phosphate group and also the CAMP. Okay, as I mentioned previous class, you need to know that the phosphate is important for the activation of this protein. And you can see here, this is the cyclic AMP dependent protein kinase. From the adenylate cyclase, the protein can produce the protein kinase and increase the activity of these protein kinases. Okay, this is through the CAMP dependent. It means that they need the CAMP for the next step. If the adenylate cyclase is not available, it's no way that you can continue with the process. Okay, you cannot process the information provided by this hormone epinephrine. So you need to have the CAMP as one of the prerequisite of the activity. This is compulsory and required. That's why we call it CAMP dependent protein kinase. Protein kinase cannot be activated without the CAMP. Once the protein kinase being activated, the body will then start to increase the activity of the certain other enzymes. This include the phosphorylase kinase or glycogen synthase. If it's increasing in activity, the phosphorylase kinase come into action, and then the calcium will start to go into the cells and increase the calcium, what we call increase the flow of calcium into the cells and start giving the activity. On the other hand, okay, the cyclic AMP dependent protein kinase also can activate the glycogen synthase for a certain activity. Okay, If the activity is to decrease the activity of glycogen synthase, they will convert the ATP to the ADP. All right. But if the activity or the carbohydrate metabolism continues, they will start to increasing the influx of calcium into the cells through the calcium ion channel. And after that, you have the phosphorylase kinase with the phosphate group attached. Once you have this phosphorylase kinase or the activated phosphorylase kinase, this enzyme can catalyze the formation of the glycogen phosphorylase. And also at the same time, they can convert the ATP to ADP. Remember when this ATP being converted to ADP, okay, adenosine triphosphate to adenosine diphosphate, you have another phosphate group, okay, which is here, and they can facilitate the degradation or metabolism or catabolism of glycogen. All right, so this is how using the hormone epinephrine you can degrade the glycogen. Right? What is glycogen? Why the glycogen is important in this hormones control carbohydrate metabolism? You need to tell me. Okay, you need to find out why. What is glycogen? Why glycogen is important when the epinephrine bound to the receptors? Okay, so that's one of your tasks. You need to tell me. Okay, what is the function of glycogen? But please do not confuse with the glucagon. All right, glucagon is another molecules. They act on the liver to increase the availability of the glucose. In other words, the glucagon is a peptide that contains 29 amino acid residues. They are being released by the alpha cells of the pancreas. Okay, but the beta cells of the pancreas produce the insulin. Okay, the insulin functions to take up the glucose and reduce the glucose level from the bloodstream, but the glucagon other way around. Okay, the glucagon functions to increase the glucose concentration inside your bloodstream. Okay, if you feel tired, you don't have enough energy, maybe your pancreas from the alpha cell of islet of Langerhans start to produce the glucagon, and then you will have a normal glucose level. Okay, we have discussed this in detail regarding the insulin, and the glucagon is other way around. Okay, they also involve the G protein to start the amplification of the signal. Okay, so this is about the carbohydrate metabolism. Okay, how the hormone controls the carbohydrate metabolism. Okay, are you okay so far? Do I have more time, or you want take 5 before we continue? I think we have another maybe 10 slides to go. I'm not sure. Let me check. Yes, we have around maybe 10 slides to go. You want to continue, or is it maybe another 20 minutes? You want to continue, or you want to have a break? What do you think?
I think take 5, Doctor.
Take 5, yeah? Okay, after 5 minutes, we continue, yeah? You just stay on the line. Okay, can we continue? Let me share the
Doctor,
screen again. Yes.
I'm sorry, but section 2, we have class at 10 a.m.
10. Okay, I'll try to finish it quickly. Okay. Let's
Okay.
see if we can continue. Okay. Okay, we just go quickly. Some of you have a class anyway. So this is as opposed to the one that we have learned before. This is the way that the glucagon can come into action. Okay, so you see, just now we learned about the epinephrine, how it can facilitate the breakdown of the glycogen. Okay, now it's the glucagon. Okay, the glucagon now functions to increase the glucose level inside your bloodstream. So what happens that glucagon can bind to the receptor, and again it activates the G protein, and also G protein will later activate the adenylate cyclase. Again, they will go through to the CAMP dependent protein kinase, and this is happening in two different ways. It's involved the enzyme fructose bisphosphatase 2 or phosphofructokinase 2. Okay, it's quite difficult for you to understand these two things, but just to make sure that you know that this is only two kinases that involve after the CAMP dependent kinase are activated. In the case of increasing activity, the fructose bisphosphatase kinase can lead to the low F26P. F26P is the fructose 2,6 bisphosphate. Okay, so this is the outcome, the molecules, not the enzyme, right? So this is the molecules, and also at the same time, the phosphofructokinase 2 can also contribute to the lower amount of F26P molecule. And F26P molecules is important to determine whether the glucose level in the body can be increased or inhibited. All right, whether the glucagon can leads to the formation of a new glucose molecules, or we call it gluconeogenesis, or they can inhibit the glycolysis. It means they can prevent the broken down of the glucose molecule. So basically, this is the images to show the action of the glucagon involving these two important kinases, okay, after the CAMP dependent protein kinase. All right, if I ask what are the important essential, for example, protein kinase to facilitate the conversion of the to facilitate the activity of glucagon, so your answer would be phosphofructokinase 2 and fructose bisphosphatase 2 enzyme. Okay, this is an example of the insulin. Insulin is a very a short change. It's only 51 amino acid in total. You see in brown on the left hand side. Okay, the rightmost left hand side here, and the pink one. So there's a combination of two chain. The first chain on the left is 30 chain. Okay, from 1 to 30 plus 1 to 21. So in total, insulin is 51 amino acid chain. Okay, they have the combination of these two combined by the disulfide bond. Okay, the disulfide means SS. Okay, two sulfur group. So they will attach these two main peptide chain and form the insulin. And obviously, during the production of the insulin hormone, they don't straight away become an insulin. In fact, they are very long. They have a 86. All right, with this blue one. Okay, so from 1 to 21, after that the pink. Okay, after that the blue one, 65 to where is it 31, and after that 30 to 1. Okay, so the connecting peptide in between in blue, all right, during the pro-insulin stage are eliminated. All right, so pro-insulin is the intermediate or earlier structure before they're being converted into the mature insulin that can be used for uptake of the glucose. Another action or similar like the previous one, you have the insulin. I have discussed this, but this is just telling you where are the subunits. Okay, this is the insulin receptors. They also activated the beta subunit of the insulin receptors, and that's on the tyrosine kinase dominant. And after that, they can give increase in the intracellular insulin concentration. This is another perspective where they involve the transporter called Glute4 Transporter. The functions of the transporter is to allow the to allow the glucose to enter the cell, right? Once they have information from the hormone receptor complex, then the cell know that the GLUT4, the glucose 4 molecules need to help, all right? They need to bind to the plasma membrane surface and then allow, open or make a new door for the glucose molecule to enter the cell. They cannot enter the cell through the same insulin receptor, no. The glucose have their own special door or own special channel. And this is what we call the glucose transporter 4 channel. Okay, and after that, they will close it. All right, so they can open and close that one. And this is also the activity of insulin. There's been lots of insulin. Maybe one of your final exam will come from the insulin part. I think in the context of the activity. All right, so this is the explanation. You want to continue with your readings, and you can see where does this insulin act on. Okay, it can act on brain because the brain needs energy, need glucose. All right, a muscle needs glucose, the adipose tissue needs glucose, the pancreatic islet also needs glucose, and after that they all will be metabolized in the liver. So the liver is the metabolism organ. All right, so they can metabolize and facilitate all this activity of the enzyme. I list down all the effect of insulin on metabolism. Maybe you want to read through. Okay, if what we call the metabolism process, right? Where does it happen? What is the outcome, and what are the molecules or enzyme involved in this metabolism? Regarding the type 1 and type 2 diabetes, is quite different because type 1 is a genetic disease. It occurs when the muscle was unable or the cell unable to use glucose to do very low insulin, and it happens in the pediatrics, in the kids. Okay, it's a genetic disease. The pancreas itself is incapable to produce enough insulin. Okay, and this occurred due to the genetic reason. But type 2 diabetes, where most Malaysian or adult have, they are due to the lifestyle, and they occur in the adult, most of them. All right, that is where the insulin is no longer effective. Okay, we call it insulin resistant. No matter how much insulin that you been secreted by the pancreas, you will not have this glucose transported into the cell anymore. Okay, they become resistant. No more glucose can enter the cell because the insulin is now ineffective, not functioning, right? Maybe the conformation of the receptor has changed, the structure of insulin itself has changed, or the capability of the cell to respond to the signal also has changed, all right? And these are all the outcomes. What happened in the case of insulin and the sport? Okay, what happened to the glucose utilization? How do they comes back? You see the work rate when you are using the oxygen, when using the outdoor activity, the glucose strength, and also the mixed activity. Sometimes you active, sometimes you become inactive, and where you don't use the oxygen that much, or an aerobic condition. I put down all the differences between enzyme and hormones for you to look through, And it's also important for you to differentiate between hormones and enzyme. And I might might ask this again inside the quizzes. Okay, maybe next week we can have the quizzes for all these lectures last week and today. This is the summary that I have discussed with you. And I think that's all for today. Thank you very much. I think some of you need to leave. If you need the copy of this, thank you very much. If you need the list of the information, you can just take it from your group leader. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. See you later. Bye bye.
Waalaikumsalam. Thank you, Doctor.
Waalaikumsalam. Thank you, doktor.
Thank you, doktor.
Doktor.
Thank you, Doctor.